In today's teaching presentation, we're having a look at the tryptophan operon. So in particular, we're looking at what happens when tryptophan concentration is either low or high. We're first going to have a look at what happens when tryptophan concentration is low. So low intracellular concentration of tryptophan are the most common conditions for the cell. So therefore, the inactive repressor protein remains inactive because there isn't enough tryptophan to actually bind to the inactive protein to activate it. The repressor gene is expressed to produce repressor mRNA that produces the inactive repressor protein. Nothing binds to the operator section, therefore nothing blocks transcription of the 5 structural genes. RNA polymerase easily binds to the operator section and it then moves along the 5 structural genes and produces the mRNA. This mRNA will then be used by the ribosomes to produce the enzymes that will then produce the tryptophan or amino acids. Now we're going to have a look at what happens when tryptophan is in high concentration. So this means that there are many tryptophan molecules, so therefore some of these will act as co-repressors and they will bind to the inactive repressor protein, activating it. So the 3D shape of the activated repressor protein changes and it binds to the operator section. This inhibits the RNA polymerase from being able to bind to the operator section, so it prohibits the transcription of the 5 structural genes. Therefore, no mRNA is synthesized, so therefore no enzymes and tryptophan amino acids are synthesized. So when in excess, no more tryptophan is produced as it is a waste of energy and the cell must conserve that energy.
In today's teaching presentation, I will be discussing about turning the operon on and off. So what does the operator do? This stretch of DNA is recognized by a regulatory protein known as the TRP repressor. When the repressor binds to the DNA of the operator, it keeps the operon from being transcribed by physically getting in the way of the RNA polymerase, the transcription enzyme. The TRP repressor does not always bind to DNA. Instead, it binds and blocks transcription only when tryptophan is present. When tryptophan is around, it attaches to the repressor molecules and changes their shape so they become active. A small molecule like tryptophan, which switches a repressor into its active state, is called a co-repressor. When there is little tryptophan in the cell, on the other hand, the TRP repressor is inactive. This is because no tryptophan is available to bind to and activate it. It does not attach to the DNA or block transcription, and this allows the TRP operon to be transcribed by RNA polymerase. In this system, the TRP repressor acts as both a sensor and a switch. It senses whether tryptophan is already present at high levels, and if so, it switches the operon to the off position. This prevents unnecessary biosynthetic enzymes from being made.